So recording has started. So in the previous class, we looked into some of the techniques that people are using to make the artificial ECM, or also known as the scaffold. And you need scaffold onto which you will be seeding your cells and then allow the cells to grow and make a mature tissue. And we looked into few of the traditional techniques like solvent casting and porogen leaching, then electrospinning, freeze drying, and also how it works, what are their advantages, disadvantages, and we also looked into few of the advanced techniques that is being currently used, like it in past 5-10 years. And one of those technique is 3D bioprinting. Okay, and we already started with that topic, 3D bioprinting. We discussed like what is 3D bioprinting. So it is nothing but it is taken from the 3D printing field, right, where you make an ink, but here you will be using a bioink, and bioink is nothing but a polymer solution, and it is mixed with the cell of your choice. And the most of the time we use a hydrogel based polymer to make a bioink. Okay, so we were discussing how to make a bioink, but before you start making a bioink, you have to do pre-processing steps, like you have to make a CAD model. You have to, if you are planning to transplant your construct on the patient, so first you have to take images of the site, and you can use X-ray, MRI, etc. And then from those images you have to construct a 3D model and do then slicing, and load your CAD model in form of STL file into your 3D bar printer. So once this step is done, you have to then prepare a bioink. Okay, so and using which you will be actually making that 3D model that you have just loaded. So from preparing bioink we discussed in brief, and today we will continue from that. So when in this slide I'm trying to explain that when you are going to mix cells with a polymer solution, generally it's a hydrogel solution, so you have to keep in mind that the polymer chains, there exists some entanglements in between them. So entanglements can be due to various like non-covalent bonds like hydrogen bonds or van der Waals interactions, etc. Okay, so you have to break them. If you don't break them, then what will happen? Then you won't be able to mix your cells homogeneously. Some part of the polymer solution will have the cells where the rest is like blank. So when you actually use this kind of bioink to print it, you won't be able to get a uniform distribution of the cells within your solid structure, because ultimately after the bioink falls on the substrate and makes layer by layer a 3D structure, you have to cross link it. Means you have to make convert it into a solid state so that its integrity remains for the subsequent tissue maturation process. So, and once the solid structure is prepared, you cannot reverse the cellular arrangement within the structure. So, while bioink, we have to make sure that the cells are homogeneously dispersed. So, how can you do that? You have to like partially break these chain entanglements. Okay, you cannot go on and use additional treatments to break all these entanglements, because because of these entanglements you will be able to get your final solid structure. Okay, so these entanglements are generally temporarily when it is in solution state, and when you add a cross-linking solution or like a photo initiator in form of photo initiator and then light, then what happens? Covalent bonds starts forming in between the polymer chain, and that results into a solid structure. So you don't want to completely break the entanglements also, you do not want all the entanglements to be present. You somewhat want to break the entanglements to such an extent that allows your cells to move freely within the polymer solution, and then you will be able to get a bioink with uniform distribution of the cells. And when you print your 3D structure and see it under microscopes, like say you tag your cells with some kind of fluorescent dye, and you observe it in under a fluorescent microscope, then you will be able to see the uniform distribution of cells, like the cells will light up. Okay, like if you are making a grid kind of pattern, the cells will be distributed in the grid. Okay, so the same thing is being written. So what people do to partially break these entanglements is they use systems that mechanically steers the cell suspension within the polymer solution. So you can use like two syringes, okay, and connect these syringes. So syringes, you can press the solution back and forth, okay, means and mix it. So you're mechanically mixing it. Or what you can do, you can take a beaker within which a polymer solution is present and add your cell suspension dropwise, and take a tip or any small thing, okay, and a glass rod maybe, and stir it gently, your cell suspension within the polymer solution. Or if your volume of polymer solution is very high, then you can actually use mixers, like where stirring system is there that gently mixes the cells. But you have to make sure that while mixing, no bubbles gets formed, okay, within your polymer solution, because usually polymers, these kind of solutions, they are highly viscous. Okay, so and after like bubbles get formed, because of this high viscous nature, it becomes difficult for us to remove those bubbles later. So you have to pay attention that bubbles are not formed within your bioink. And if the bubbles get formed, then it can either clog your nozzle, or you won't get a 3D structure that is continuous in nature. Rather, you will get a 3D structure with bubbles in it. Okay, and that will again like hamper the distribution of oxygen and nutrients later on. Okay, so you don't want to create any bubbles while mixing your cells with the polymer solution. Also, you have to make sure that after the 3D structure gets formed, okay, you are exposing it to some secondary cross-linking system, like using a chemical or using light or something like that to create covalent bonds. So you have to make sure the degree of cross-linking, okay, how much is it, you have to optimize that. If your cross-linking is too much, then the cells won't be able to move around, because the pores within the 3D structure will be too small within which the cells will move. And if you're cross-linking it very loosely, then the mechanical strength of your structure will be very less, right? And it will start breaking off, or after a few days of tissue maturation. So this degree of cross-linking is also something that we need to make sure of while you are making a polymer solution, because depending on the concentration of the polymer solution, also the concentration of your cross-linking solution, your cross-linking degree will vary. So you have to keep these in mind, like how much concentration to use to make your bioink, and then how much concentration of cross-linking solution or photoinitiator, etc., to use for cross-linking to get a final 3D shape with cells encapsulated within it, and they should be able to get nutrients and oxygen properly and move around within the 3D structure. Okay.
Ma'am, you said we should also not break all the cross-linking, right?
Yes.
But how will we know this much cross-linking is all present in the bioink?
So what I have observed during my work is, if you are using a very high concentration of polymer, then the chain entanglements will be high. How will you know that? You will be able to see the viscosity. The solution will be highly viscous. Means if you take your solution within a tube and try to turn it upside down, and note how slowly your solution is trying to move towards the opposite end. The highly viscous solution, it will take more time. Okay, so it means your entanglements is very high. So you can either use a then go to a lesser concentration, so that like entanglement is less. Okay, and then compare in between the two polymer solutions how the cells are able to distribute. So you can try to make bioink using both the solutions. Okay, and print a 3D structure and then observe under microscope through like some fluorescent dye, etc., and see how the cells are distributed within the hydrogel. Okay, so if you see uniform distribution in the lower concentration polymer, then you can assume that your bioink was successfully prepared with this percentage of your polymer. And the second one, if the distribution is not good, then you can assume that you couldn't make a uniform cell suspension or homogeneous cell suspension within your polymer solution. Okay, so that this kind of small experiments will help you optimize the like composition of your bioink. Okay, so what are the factors that influences your bioink preparation? The first thing is cell density. So there you should give a limit to how much cell density you can work with. Ideally, we want like the cells to remain in the range of 10 raised to power 7 to 10 raised to power 9, because that is the clinically meaningful cell number, right? Like, and it correlates with the cells number of cells within a tissue. But within in the bioprinting field, people usually stay up to 10 raised to power 6, because if you use a very high cell density, then the cells usually like interfere with the cross-linking process. They don't allow proper cross-linking within the polymer solution, within the polymer chains. So the mechanical strength of your structure gets compromised. And when people are studying these tissue constructs for say 20 days, 30 days, and then also doing animal experiments, they don't want this to happen. Okay, they want the mechanical strength to like sustain as long as possible for their experiments. So currently people are working with very high cell very low cell densities than what is needed. Okay, then although people have tried like achieving very high cell densities using scaffold free bioprinting. So what is scaffold free bioprinting? Here you don't use a polymer solution. Okay, your bioink is just a cell suspension where cells are suspended in a media. So in that case, high cell density can be achieved. But again, the limitation is the type of structure that will be made from this bioprinting approach. It will have a very low mechanical strength. Okay, so you cannot really translate these kind of structures for transplantation purposes. It's good for screening purposes, like you can make spheroids, tumoroids, etc., out of out of this technique, like using scaffold free bioprinting, but for the purpose of drug screening, right? But you cannot really use these kind of structures for transplantation or in vivo studies, etc. Now, depending on the printing technology that you are going to use, cell density will be affected. Okay, so if you are using uh, print technology where your bioink is going to be extruded out from a tiny nozzle, then the cells will be exposed to very high shear stress, because of which there will be loss in cell number. Also, if you are using a technique where light source is used, okay, we are using light to make your 3D structure, then the wavelength of the light in which you're working is going to affect your cell numbers. And if you are using like inject kind of technology where drop wise your structure will be made, then depending on the distance between the needle or the nozzle and the substrate, uh, your drops can either, if the distance is too high, the drops will, rather than forming a spherical shape, it can splatter, like you have seen water droplets falling in the sink, it can splatter, no? So if that kind of thing happens, then cells can burst immediately upon this mechanical impact. So you have to optimize again your bioink properties so that your shear stress on the cells reduces, or you have to use a bioink which allows using a light wavelength which is cell friendly, or you have to optimize your bioink like composition, or you can say the properties like viscosity, etc., so that proper drops are formed, and that splattering, drop splattering doesn't happen. Then you have to make sure whatever material, say like polymer receptor, whatever material you are using for making a bioink, it is not toxic to your cells. Okay, if it is toxic, then it will hamper your cell viability. Then next thing is another factor, influencing factor, is the printability of the bioink. So what is printability? The term printability. So it basically means uh, whether the bioink is printable or not, whether uh, it can print and create some 3D structure. So people have defined how to measure printability in various ways. So you can either see whether your bioink is coming out of your nozzle without clogging it. Okay, and this is applicable in extrusion based technology or drop on demand based technology. Okay, so your bioink, if it doesn't clogs your nozzle or needle tip, then it is printable. Another way of determining whether your bioink is printable or not is seeing whether it forms filaments. Okay, filaments or thread-like structures upon applying some pressure, and then this is again applicable for extrusion-based technology. Then it should, the ink, whatever ink that you're using, bioink you're using, it should be able to create that structure, that's fine, but that structure should be stable, it shouldn't collapse. Okay, because you know the gravity is working. So if you're making a tall construct, like a few 10-15 layers of construct, then each layer should be able to support the above layer. Okay, and shouldn't collapse under the gravitational force. Okay, under its own weight, basically it shouldn't collapse. So if these three criteria are met, then you can kind of say that your bioink is printable. But there are other criteria as well, like that tells you in form of numbers that your bioink is printable or not. So one thing is determine is rheological properties. So rheology is another completely different field of chemical engineering, basically, where people study the uh, flow of non-nutrient fluid under some stress. So you apply some shear on a non-nutrient fluid, and people study how it is flowing. So, and they do various kind of experiments to determine that, and they use various parameters as well. So when you study the rheological properties, so usually the bioink is a non-nutrient fluid, that is why we are going to rheology field. So people study these properties using rheometers. It's a machine again, in form of some parameters like loss modulus, storage modulus, etc., and say that under these optimum conditions your ink is printable. Then also you have to study the gelation kinetics, means how fast your ink your bioink transitions from the liquid-like state to a gel or solid state. So if the kinetics is too fast, okay, if the kinetics is too fast, then what can happen? While printing, your ink will gelate within the needle, okay, and will clog it. Or if it is too slow, then what will happen? If you are printing multiple layers, then your bottommost layer won't be won't form a solid structure. Okay, so when the printer tries to print another layer on top of it, then the bottom layer will basically start spreading. Okay, and the second layer will also start spreading. So the 3D internet 3D structure will not form, rather you will get some irregular shape. So these kind of studies or analysis one needs to do. Then also people see the surface tension of the bioink. So if your cell density, that whatever cell number that you have used to make your bioink, if it is too high, then the surface tension will be low. Okay, and the surface tension, if it is low, then what will happen? uh, so your 3D structure basically then will start spread. Again, the same problem that I just said. It will turn into um, like puddle kind of thing. It will like for example, if you put a drop of water on a highly hydrophobic surface, then what happens? It forms a sphere, no, because surface tension is very high. Similarly, if your bioink has a high surface tension, okay, at a lower cell density, then uh, your each of the filaments, if you're using extrusion base, then each of the filaments will retain its shape properly. All right. So these are the influencing factors that um, helps you understand how good your bioink formulation is, okay, and how good are the 3D structures you can get out of from this bioink. Now, now you can understand that the term printability is a very important term, okay, and which is why people actually study this thing very in depth. People are studying this, okay. So they see the rheological properties, okay. So under rheological properties comes the viscosity of your bioink. People try to understand what is the range of viscosity at which your bioink gives very good shape. Then they use shear thinning bioinks. So it is important if you are using extrusion based printing method. So what is shear thinning? Shear thinning is a phenomena, okay, that is seen in non-Newtonian fluids. So what non-Newtonian fluids, water is a Newtonian fluid. So shear thinning doesn't happen with water, but shear thinning can be seen with fluids like honey. So if you take a bottle of honey, okay, and try to pour it on top of toast, same, then what happens when you try to pour it? So you are applying some like you can say shear force onto the honey, and honey will first form a droplet and starts to drop on your toast in form of filament. Okay, so when this why this is happening? Why this is happening? In the bottle, inside the bottle, the viscosity of the honey is very high. But when you are tilting it up in order to pour it, then you are applying some shear. So in presence of that shear, its viscosity started to decrease, and it transitioned to a liquid-like state. And because of that liquid-like state, you will be able to get your honey on the toast. And as soon as it falls on the toast, you will see that it will no longer able to flow. It will rather stay on it. You have to use a knife to spread it. Okay, so this is called shear thinning, and it is a very common phenomena in our daily life, like the food that we eat. Most of the materials that we consume, you can actually categorize them into the shear thinning class. So for the bioink as well, we want those bioink which are shear thinning in nature, especially for the extrusion-based 3D bioprinting technology. Because when you're like loaded your bioink within a cartridge, the material has a high viscosity. Okay, it will not flow under gravity. It will not flow that easily like water. But when you're applying some pressure, means the material is experiencing some shear, and it under that shear it transitions into a liquid like state. If it doesn't happen, then your needle will be clogged and you won't get filament like structures to form your 3D shape. So you want that liquid like state to happen, and it is only happening when you're applying some pressure. That means you're applying some shear force, and as soon as your bioink falls on the substrate, the shear force is gone, so its viscosity increases. Okay, its viscosity increases, and because of this high viscosity, the 3D shape that you are trying to form gets formed rather than spreading out. Okay, so that is one rheological property or that people want in their bioink for the extrusion based technology. Another thing is the gelation kinetics, means how fast your bioink is turning to a solid state, solid like state. That is also people study under rheological properties. Then people see the yield stress. Okay, how much stress, or what is the minimum amount of stress that we need to apply on our bioink in order for it to flow. It should flow, right? Means it should come out of your needle. So people also measured that. Then shear modulus. So again, from the like previous discussions when we were discussing about Young's modulus, etc. So there is another term called shear modulus, which is nothing but the proportionality constant between shear stress and shear strain. So shear modulus is also measured for the bioink to determine whether it is printable or not, and then the surface tension. Okay, that is also measured. So what are the factors that influences your 3D structure, means how your 3D structure will look like? First is, um, does your bioink layers, that if you're forming the 3D structure layer by layer, so does the layers merge with each other, okay, while it is printed? If it is merging, then most probably you won't get like the 3D shape that you intended. You would see like a lot of difference between your CAD model and your final structure. So people don't want fusion of these things. So people are working on like better bioink formulations to prevent this. Then also people don't want that under gravity your structure collapses. Okay, that is also not required. That is not also not desirable. Then if you are using droplet based technology where drop wise your structure is formed, then your drops should be formed of uniform sizes. Okay, all the time. It should not be like your a few of the drops are large, a few of the drops are smaller, then it will then hamper the cell number as well. Okay, because the volumes are different, so the cell number in each of the drops will vary. The 3D structure, you won't get a uniform cell distribution within this 3D structure. Then the concentration of the cross-linking. So whatever cross-linker that you have used in your bioink for the technology, whatever XYZ technology, you have to make sure that their concentration should not be too high or it should not be too low. It should be at the optimum level so that the pore size allows cell infiltration and cell migration properly within the 3D structure. And then solidification means how fast or how slow your 3D shape turns into a solid structure. Okay. Now we have discussed a lot about the bioink preparation part, right? And we have briefly discussed that there are three technologies basically that exist in the bioprinting field. So one is the extrusion based, another is the droplet based, and third one is the light based. So let me just show you small clips of like each of these types. Okay, small video clips basically, so that you will be able to visualize what happens when I'm talking about extrusion based 3D bioprinting and others. Okay, so I don't know whether you will be able to hear the sound or not, but um, so so this is an extrusion based 3D bioprinting going on. First watch this video, then I'll explain what is happening. Are you guys able to see the video?
Yes, ma'am.
All right. So again have a look. So what we understand from this video is, in extrusion based 3D bioprinting, there is a needle, okay, and the cartridge. Cartridge is basically the small container which holds your bioink, and this bioink within the cartridge is like pushed out, like some pressure is applied, because of which this cartridge, out of this cartridge through the needle, your bioink comes out. So as it comes out in form of filaments. So in this video you are seeing that these filaments are coming out in a circular shape to, and it has formed multiple layers. Okay, and these all these layers have like together formed a tube like structure. So that is extrusion based. So there is a needle involved through which your bioink will be uh, like on application of pressure it will come out, and it will come out in form of filaments. Okay, and using these filaments you will be making a 3D structure. This is the extrusion based 3D bioprinting demo video. Next is the droplet based. Okay, so see this. So are you able to understand what is happening here? The video is clear, no?
Yes, ma'am.
The ink is dropping from the cartridge, ma'am.
Yes. So this printhead, okay, this printhead has the cartridge, okay, and at the bottom of this printhead there is a small aperture through which drops are like coming out, and through drops only the square grid kind of shape is formed. All right, so how people make 3D structures out of it, you might wonder. So they use something called another support in form of biopaper. I have seen in literature. So one layer of on one layer of biopaper, these droplets containing cells are like put in a line, like in a geometric fashion, like either in grid form or whatever design they want, and then top of it another biopaper is like kept, and again on top of it the drops are formed. In this way, in between two biopaper the drops are present. Okay, and like either through cross-linking mechanism, some these drop biopapers are cross-linked with the cells containing drops in between. Okay, and in this way they make the 3D structure. So that is one way. Another way basically to make a 3D structure using the droplet based technique. And another video that I have for you is where they are using light, okay, to 3D bioprint a structure. Have a look. So did you see what happened here? The 3D shape actually. So what is happening is this particular light based 3D bioprinting technique is called digital light processing 3D bioprinting. I'll come to it obviously in the upcoming slides. So here what is happening, the uh, top the top structure which is moving up and down, okay. So, and in the bottom there is a petri dish with your bioink, and this bioink is light sensitive, means in presence of light this bioink cross links. So as one layer gets cross linked, it adheres to the top platform, and this top platform then moves a little bit up, and then the next layer, for making the next layer, another like a small layer of bioink gets cross linked, and it gets cross linked to the layer adhered to the up and down, like the silver colored bob. And in this way the structure actually gets formed in an upside down manner. So the lowermost layer gets formed first, and then the like top layer. Okay. So and when the total structure is formed, you have to just remove it from the silver head. Okay, and then do further processing. So this is another demo where you have also seen some flashes of light continuously coming. So what this light is doing, it is cross linking each layer. First layer is formed, it is adhered to the silver head, then the bioink comes, forms a thin film, it gets cross linked and adheres to the layer that is previously formed, and in this way subsequent layers get cross linked individually by the light. That's why it is sparking. And it is this kind of technique gives very good resolution structures. So here actually they made a knee meniscus, okay, and they compared it. So they compared it. So this is the yellow knee meniscus is made from the DLP based 3D bioprinting, whereas the like transparent meniscus they made it from maybe extrusion based. So you can see the resolution of the structure is better in the light based technique. Okay, so now you can visualize when I'm talking about each of the 3D bioprinting techniques. Okay, because briefly I will discuss what is happening here. So this is another image basically that again categorizes each of the above techniques. So when you're talking about material extrusion, like extrusion with 3D bioprinting, extrusion can happen uh, either by applying pneumatic, either applying air pressure, that is pneumatic extrusion. So some compressed air is used to push out your material out of the needle in form of filament, or there can be a screw or piston which is directly in contact with your material to extrude it out. Okay, so these are the two ways, or if we include the screw one as well, the three ways through which you can extrude your material in the extrusion based 3D bioprinting. Then in the droplet based 3D bioprinting, where the drops are formed based depending on what type of technology 3D bioprinter used to form the drops, you can again categorize it. So they can be thermal inkjet. So there is small heater that heats up your bioink. So in presence of heat, what happens? Its viscosity gets reduced, and it becomes more liquid-like. So it easily forms a drop. So that is thermal. It can drops can form in presence of piezoelectric circuits, something like that, or in presence of like electrostatic plate, or means electric field, or microvalve, acoustics. So these are, and also lasers are also used, like laser induced forward transfer. These are all the technologies that may be present in a 3D bioprinter using which your drops are formed in aligned manner, like in whatever geometry you're looking for. Okay. And the technique that I just showed you the last video, the DLP1, that is called digital light processing. So that is one of the technology that comes under Vat Polymerization Based Bioprinting. Okay. So here, but this entire block comes under light based 3D bioprinting. So here basically you use a bath containing photo cross linkable bioink, and depending on how the light falls on your bioink, okay, and how the stage, is it moving up, like where it is present and how it is moving, you can categorize them into these three types. Okay. So let's go into slight more detail. So extrusion based 3D bioprinting, it is one of the like most used, like most one of the widely used method basically. Okay, means most of the publications you will see people have used extrusion. Okay, it is easy to understand the parameters to optimize for extrusion based bioprinting is relatively easy, because most of the literature you will find on the extrusion based 3D bioprinting. Then here what happens, as I told you, the material comes out of a needle or a nozzle on application of a pneumatic force, or pneumatic pressure, or you can use a mechanically you can give some pressure using a screw or a piston. And for the extrusion based bioprinting, you need the bioink to be shear thinning in nature. All right, I already explained why. Then it has some advantages, like you can create very small structures or you can create very big structures. So it gives you the advantage of scalability. Okay, and uh, you need a bioink with a high viscosity. Okay, and the structures that you finally print, you will be able to achieve high cell density as well at some optimized conditions. But there are some disadvantages as well. The resolution of these structures is low. So by resolution I mean, if you compare your 3D structure made out of extrusion based 3D bioprinting with your CAD model, you won't see that they like both 100% like similar. Okay, there will be like large differences in between those two structures. And the lowest level of lowest diameter of filament that you can go in extrusion based 3D bioprinting is in the microns level, like 100 or 200 microns. You can't low you can't go beyond that. Okay, so lower than that. So the structures will have quite thick 100 micron thick diameter is quite thick actually. So and the space between the two filaments you won't be able to see, they will most likely merge. Okay, if you're using a pattern where the space is less, and you won't be able to differentiate between the two filaments that well. That is why I'm saying that it gives a low resolution 3D structures. And also, since this technique requires the use of highly viscous bioinks, and if your parameters are not optimized, so most likely during your initial trials you will see nozzle clogging happening. If your nozzle clogs, then your just robotic arm, it will just move without dispensing anything in the bottom. So because that means your nozzle is clogged. Then I already explained that extrusion mechanisms exist, like you can use a pneumatic system where air pressure is used to push out your bioink out of the nozzle orifice, or you can use a piston or a screw rather than a pneumatic system to apply pressure on your bioink. Okay, so but it has both has advantages and disadvantages. So a few bioprinter uses the pneumatic system, few bioprinter uses the piston or a screw based system. Okay, now the droplet based bioprinting. Okay, so I'm going a bit fast here. So if there is any query so far, understanding anything, like are you able to are you able to follow along with me?
Ma'am, you talked about laser bioprinting, right?
Yes, not laser light. Yeah, yeah.
How laser light will help us here, ma'am?
So can you refer to the slide, like from where you got this query?
The picture you showed 2 slides back.
Here,
This slide, here, ma'am.
this one.
Yes, ma'am.
So, like here, what is happening is, so laser, you can actually focus. This is a very focused beam of light. Okay, so you can control it where the light falls very accurately. Agreed. So what happens is you use a support layer, all right, and there is another layer which absorbs the excess laser energy, and in bottom of it your bioink is present. So, and on the support layer you apply your laser. So wherever the laser beam, you're like targeting, wherever your laser is going to fall, your bioink that is there at the bottommost layer, it will come out. Okay, and it come out and fall on the substrate in form of a drop. So in this way they are like monitoring, or basically precisely controlling the arrangement of the droplets of bioink on the substrate using a laser. Okay, is that all
Okay,
right?
ma'am.
Okay.
Okay, ma'am.
So I already discussed about extrusion-based 3D bioprinting. Now, briefly, I'll be discussing the droplet-based bioprinting. So it's again a very popular technique. In fact, the first 3D bioprinter that is reported in literature is the one where the researchers have actually modified a commercial inkjet paper printer, like in the offices that we use inkjet printer. So they have actually modified that inkjet printer to print cells and show that it is a and give the proof of concept of this technology. So that is why it's a like very popular. So people actually started with droplet based technique and then moved on to extrusion and other techniques. So here what happening here is you're using a nozzle or a needle, or in case of laser based you're using a laser to accurately position your droplets of bioink in form of some pattern. Okay, and if you want to create some 3D shape, you use a support structure like a biopaper, something like that, to build onto your 3D structure. Okay, and like depending on the technology that you are going to use or your 3D bioprinter is using to form the droplet, you can again categorize it, the droplet based 3D bioprinting in form of like inject. So in this diagram, in form of thermal inject bioprinting. Okay, then piezoelectric, electrostatic, hydrodynamic, electrodynamic, acoustic, then microvalve based. So depending on that, you can further classify your droplets with 3D bioprinting. And please remember that each technology, like even the classification that I'm mentioning here, where did it go? So even the classifications that I've mentioned here, you have to again optimize each of the parameters, like how much, if say in thermal inkjet, you have to optimize what is the best, what is the maximum temperature you can go up to, okay, without compromising your cell properties like its viability, etc. Then in piston extrusion, depending on the bioink formulation, you have to optimize how much pressure is best suited for printing. Then in the laser based, you have to optimize your strength of the laser that you are going to use for this application. So all these parameters are different for each of this technique, but your ultimate goal remains that the droplets should be of uniform size, so that each of the droplets contain approximately equal number of cells in it. All right, and ultimately your bioink material, if you're using some polymer, then that polymer should support your cells, like they should not cytotoxic, right? Then the bioink properties, like the viscosity, etc. So those things you need to optimize depending on what technology you are going to use. So I'll just briefly discuss about the inkjet bioprinting. So here, what happens, you can either continuously add drops onto the substrate, or you can do drop and demand. So your machine can like support continuous, like as soon as you start applying pressure onto the cartridge, from the tip of the nozzle, the droplets, a string of droplets starts forming, and it deposits onto a substrate, printing substrate. It can happen like that. Or what can happen, you can program your machine to uh, accurately apply pressure only at the point where a droplet is needed. Otherwise, the pressure will not be applied. That is called drop on demand. So both of these techniques are useful to create complex structures of your desired application. Okay. So there are some advantages basically of droplet based techniques, like it will help you get very high resolution. So in between two droplets, you can within a layer, you can actually understand that these two droplets are distinct. There's some space. So visually you can understand that space. So there is that high resolution thing coming. Then printing speed is very fast. So within few seconds you will get one layer ready, then the next layer, the next layer. So very fast printing is possible. Then affordable. This, since I told you, the first bioprinter is the inkjet, basically the modified the inkjet printer that is used in office. So the principle remains the same. The cost is very much low. So droplet based bioprinters are very much easy to procure and much cheaper. All right, to start, if anyone wants to explore 3D bioprinting and they don't have much money to start with, so they can definitely purchase a 3D bioprinter that is based on droplet technology and start their work. Also, uh, if someone's assay or experiment desires to study the effect of some chemical on cells of different concentrations, like cells of different densities, then they can introduce that. So depending on the size, they can vary the size of the droplets and then vary the cell concentration and create a gradient, cellular concentration gradient, and do their experiments. So this kind of tweaks is possible uh, depending on what application or what experiments that you have in mind. But there are some disadvantages as well. Very low viscosity bioinks can be used. So viscosity which is close to the viscosity of water, that is 1 centipoise. So you have to use those kind of bioinks. You cannot use a highly viscous bioink. So if your bioink contains some polymer, so you have to make sure that polymer is not too viscous, and the viscosity remains in the range of that of the water. And if the viscosity is high, if suppose you surpass that threshold level, then what will happen, your nozzle will again start to clog, just like we discussed in extrusion based 3D printing. And if there is clogging, then there will be discontinuity in your structure, and your final 3D structure won't be formed properly. So this was about a droplet based 3D bioprinting. Now the light based 3D bioprinting. So the current technology that is there in light based 3D bioprinting is the vat photopolymerization. Just like I showed you in the video, the DLP 3D bioprinting or digital light processing. So for explaining this, let me go to this figure. That will help you understand what is actually happening again, like, or if you already understood from the previous explanation, that is good, but if someone has still any doubt, they can, that those doubts can be clarified now. So under Vat Polymerization Based Bioprinting, we have again three bioprinting techniques. First is the stereolithography, next is the digital light processing, another is the two photon polymerization. The common thing among all these technique is a light source should be there. It can be either UV light or visible light, okay, depending on your machine configuration. But what is varying in all these three? Another common thing is there is a bath or a vat containing a photo cross-linkable polymer. So these two things are common in all three like techniques. But only thing that is varying is the component in between the light source and the vat. What is that component? So in stereolithography, there is a scanning system. So in these are all 3D bioprinters. So you have to first give a CAD model to the machine. Okay. So and the machine is configured to read that CAD model. But how it reads and translates it, that technology is varying. So in stereolithography, there is something called scanning system. So it basically scans the CAD model, slices it into different layers, okay, and the light that is falling on the system is made to fall in a distinct pattern depending on each of these layers. So the light that will fall on the vat, it is falling in a pattern way. It is not falling just like a ray of sunshine. It is not falling like that, but it is patterned. So as this stage is moving, one layer gets formed depending on the first layer of the CAD model and depending on how the light fall on it. Then again the stage moves little bit up, the bioink comes on top of it, and the second layer is formed. Where digital light processing, here again the mechanism of how the light falls is changes. Here there is something called digital micromirror device and imaging optics. So here this machine is capable of making like you can say patterns where some regions are dark, means light will not be present, and some regions are light. So this machine can do, only you have to do is supply it with the CAD model. The processing part to make this kind of patterns the machine will do. The based on this pattern, the way the light falls, like you can see the light is falling on this imaging optics and two rays got formed. So that this means that the pattern where there was dark regions, the light was not falling. So in middle there was no light, whereas where the light is supposed to fall, the lighter regions, the light will pass through them. So in this way, the way again the light is patterned in this technology, in this unique technology, in this using digital micromirror device, and the scaffold is formed in a layer by layer fashion as the um, thin layer of bioink comes on top of the previously formed layer. It gets cross-linked and then it stage moves up. Then again another layer of bioink falls on the previously formed layer and it get cross-linked. This way layer by layer cross-linking as well as assembly of the structure takes place. Whereas here in two photon polymerization, we are simply using a objective lens like the lens present in the microscopes to uh, position the light where it falls in the vat or the bath containing photocross-linkable polymer. All right. So that's that's it. Maybe we can discuss some advantages, disadvantages. Yeah. So the advantage of using this kind of light-based 3D bioprinting using a bath of photo cross-linkable bioink is you will get a structure which is having a very high resolution. So you can actually distinguish between the two layers, or you can distinguish the pattern, like where there is an empty space or where the space is completely filled. So this kind of distinction is possible within these structures. The process is very fast, it happens very quickly. Actually, you saw right in the video that although that video might be uploaded at a high speed, means 1x or 2x speed, but still the process is quite rapid. So you can make even relatively big structures, like the size of a small golf ball you can make, and that will take hardly few minutes. Disadvantage is uh, the bioprinters using this technology are very costly in comparison to the extrusion based and the droplet based, which is why very few labs in the world are actually working on this technology. And cells are often sensitive to the UV light, means you have to be very careful with the light wavelength that you are using. Okay, and the time for which that light is exposed, like the cells are exposed to that light, because it can again compromise the cell viability within your 3D structure. So that's it uh, about 3D bioprinting in today's class. I would like to know if you have any questions. Are there any?
Ma'am,
Yeah.
you said right, ma'am. We check viscosity and some other factors, right? After preparing our bioink, uh, we will use any instruments to check those things or
I,
manual techniques,
I
ma'am.
can I cannot hear you well. Actually, your connection is poor, I think. Can you repeat your query?
Yes, ma'am.
Can
Once
you repeat?
we prepare our bio, yes, ma'am. Yes, ma'am. I'll repeat, ma'am. Are you hearing me now, ma'am? Am I audible,
Yes,
ma'am?
yes, I can hear you.
Yes, ma'am. Once if we prepare our bioink,
Hmm.
does we use any special instruments to check those factors you listed down, like viscosity, stress, shear stress, strain, and all? Or by practice we will come to know, or by the result we will come to know?
So, for we have to actually measure those characteristics. Like we have to determine the viscosity. Then we have to do experiments to see whether your bioink is shear thinning or not. Okay, and also we have to do experiments. And
Oh, that's what, ma'am. What are the experiments we do?
also,
Just a hint, ma'am. That's what.
so I don't really. Biology, there are
Okay, ma'am.
some experiments like the flow sweep experiment, that within a repeater you load your sample and allow the
Ma'am, you're breaking, ma'am. You're breaking.
hello, am I audible properly?
Now it's possible, properly.
Okay. So as I was saying, there is an experiment called flow sweep experiment, where you like using a rheometer. It's a machine. So you load your sample into a rheometer and allow and program your rheometer to give continuous shear onto your sample. And as the machine is providing shear on this sample, it will measure the changes in viscosity. So in this way you can understand whether with increasing shear your viscosity is declining or increasing. So this is one experiment that you can do with using a rheometer, that is flow sweep. Another type of experiment can be determine how the structure of your polymer or your bioink changes, um, like whether it transitions to a liquid like state. So if it does, then people measure its loss modulus, which is G double prime. It is represented with G double prime. And if it is if it attains a solid like structure under some condition, then people measure its G prime. So people then see how these two factors, G prime, this G prime is storage modulus, the storage modulus and loss modulus, how these two parameters varies on, say, as a function of cross-linking solution, okay, or as a function of temperature, means as a function of the stimulus that you already know is going to cross-link your material. So in this way you can understand how the polymer or bioink is transitioning on application of the stimulus for cross-linking your like final structure. Okay, there are other experiments as well to know more details. So these are some rheological experiments. You can do like yield stress experiments as well using a mechanical testing machine. Again, you have to then apply stress, you have to apply some strain, correspondingly some stress will be generated, and you'll see at what point that linearity between stress versus strain is no longer present. It will like starts to flow. Okay, means you will see some curve rather than a linear line. So at that point you will be understand that this is the amount of stress needed for the material to flow. So this kind of, so these are few examples that I'm aware of or have used in my experiments. There are others as well, maybe you can check it out, and if you do find, let me know. All right. Yeah.
Okay.
Yeah. Oh, I forgot to mention that you can also measure the surface tension using contact angle experiments. Okay, so that is another class of experiments where you can see whether your bioink is more towards hydrophilicity or hydrophobicity. Okay, and what is the surface tension values and all. So that is another experiment that is there. I cannot really recall the name of the instrument being used, but if you just simply search contact angle measurement, you will be able to find that instrument's name. Okay, because that instrument is only used to measure the surface tension. Okay. So I was thinking to like stop today's class now and continue maybe on Thursday, because I'm going to start another topic within the biomaterials section, which is the delivery of signaling molecules. Basically, the molecules that that is going to guide the behavior of the cells within your 3D scaffold. So up to now you understood how to make a scaffold and how to load cells into your scaffold. But if you want some special kind of cellular behavior, okay, or you want to observe some unique function specific to an organ, then you have to like modulate those cells. And how will you do that? For that you have to use the signaling molecules, and you have to deliver those signaling molecules at the appropriate target site so that it reaches your cells of interest and shows the desired response. So I will discuss this topic. I'll start this topic in the next class. How about Thursday? Does Thursday sounds good? Like from 7 to I think 8, a 1 hour or maybe 1.5 hour class will be there. So the sound Thursday sounds good. Yeah.
Yes, ma'am.
So we will then meet on Thursday, that is on 16th at 7 p.m. Okay, and we'll start this topic. All right. Okay. Uh, Let me also stop the recording.